This is what DNA looks like when it is found in its natural state. It'll form a double helix structure where one strand of the DNA will ravel around the other. And this is what DNA looks like in its unraveled state to be able to better describe its properties, features. Starting off, we have the phosphate deoxyribose sugar backbone right here, one on each side. And what makes this backbone is the sugars, the deoxyribose sugars, and the phosphates. And it is a chain of those that creates the phosphate backbone. Now, the sugar is what binds to our nitrogenous bases. These right here are nitrogenous bases, and these are what make up the genetic information of a human, the different, or of any organism. The different sequences of these bases is what defines the properties of an organism. So for the nitrogenous bases in DNA, there are four. There is guanine, cytosine, adenine, and thymine. And in this group of four nitrogenous bases, there's two groups: purines and the pyrimidines. So the purines are going to be a little bit bigger because they are double ring structures, being guanine and adenine. So the bases are actually bigger, chemically and physically bigger. And then the pyrimidines are going to be a little bit smaller, thymine and cytosine. And how it works is the nitrogenous bases will always, two of them will always pair to the same one. So, for example, guanine will always pair with cytosine in DNA, and adenine will always pair with thymine in DNA. As you can see, this process is these bonds are replicated: guanine, cytosine, adenine, thymine, guanine, cytosine, and that process repeats in different combinations in different orders, obviously. And in these different bonds between the two bases, there are different strengths of bonds. So between cytosine and guanine, there is a 2-hydrogen bond, and between in cytosine and guanine, there's a 3-hydrogen bond, and adenine and thymine, there's a 2-hydrogen bond. Cytosine and guanine are harder to break apart. So DNA rich with cytosine, like DNA sections rich with cytosine and guanine, will be harder to break apart because of this 3-hydrogen bond. And adenine and thymine will be easier to break apart because of the 2-hydrogen. To add on, DNA will always have an anti-parallel, meaning that the 3' end will always be facing a 5' end, because the two backbones of DNA will always be facing opposite directions, which creates the anti-parallel. 5' face the 3', 5' face the 3'. This is my model of DNA replication. Starting off is the template strand, which is used to make the entire DNA replication. It is the original strand that is used to make the two new strands. And this is how semi-conservative replication works, because both sides of the template strand are used to make the two new strands. Okay, so starting off, the template strand is in its wound double helix natural form. Right here, topoisomerase will come and unwind this wound DNA. And then after that, helicase will come and unbind the complementary base pairs. And now that they are in an unbound, single strand, unstable situation, single-strand binding proteins will come and stabilize that situation so that replication can occur. So in replication, there are two different strands, and these two different strands are replicated in different ways. This is the leading strand. It is replicated continuously without any breaks or pauses with a DNA polymerase that will come in and do it continuously, replicate the DNA continuously. The lagging strand, however, replicates differently. It will replicate. It'll replicate in different fragments. So right here, a DNA polymerases will actually. This right here is the DNA polymerase, by the way. This big situation right here, and then okay, so DNA polymerase will actually come and bind different DNA polymerases will come and bind to the fragments and replicate the template strand pretty much randomly. And this will create little fragments known as Okazaki fragments, which are separated from each other. And what will happen is DNA ligase will actually come in and connect these fragments. As you can see, it's connected back here because pretend that this goes on forever and ever as an actual replicated DNA strand. DNA ligase has connected all the Okazaki fragments leading up to here. And there's no need for that here, as DNA polymerase will just come in and replicate continuously. So before DNA polymerase can work, however, RNA primase has to be this right here. RNA primase has to come through the phosphate backbones, because what it does is it creates RNA primer, which DNA polymerase actually needs and uses to be able to start replication, because DNA polymerase will bind to the RNA primer, and that's where it'll start its replication progress. And to get into more detail of what DNA polymerase is, DNA polymerase will do nucleotide base reading. It'll check for the nucleotides. It'll not only check and make sure everything's correct, it is the protein that creates the secondary strand. It comes in, it reads, okay, so guanine, boom, cytosine, guanine, cytosine, thymine, adenine, cytosine, guanine. And it'll continue to keep reading the pairs and replicating. And this process will continue. This will keep unwinding. This will keep unzipping. This will keep stabilizing. This will keep priming. This will keep replicating. And this will keep connecting.